things.
Antibiotic resistance, is that?
Yeah, you need some kind of a selection. But what usually do you use? Well, nowadays it changes. But what is the main method used for the knockout here in this case? We had it already before. It's recombination, right? And then when you have it in the cells, you expand. You select them. You expand. So then you bring back your selected cells into another blastocyst, and that you implant into a mouse. And then what? You have an embryo with a couple of cells in a mouse. How is that going to give you a transgenic mouse in the end? What happens with the embryo? Ideally. What happens with any embryo?
It develops.
They will have offsprings and next generations.
It's being born.
Yes.
Right? So you hopefully get pups. And then this pups, what do you do with the pups? Or what do you need to do with the pups?
Breed them.
And why?
Yeah.
Because the key element is still missing, probably.
To see if it will successfully pass on the trait that you are looking for.
Yes, and what's the requirement to do that?
They have to be homozygous for that?
Not necessarily, but that's beneficial.
You have to keep breeding them until there's the transmission of the new gene.
Yeah, and how do you transmit that from one generation to another one?
Breed them amongst each other.
Interbreeding.
If you have transgenic liver, will you transmit that to the next generation?
No, it has to be in our germline. So
exactly. So you need a germline event. Because the first generation, that's the key point. Well, first of all, here you have your homologous recombination that you integrate into cells from the blastocyst, into the pluripotent stem cells, and those ones you select. And then you inject them back. And after you've done that, you implant that into a pregnant foster mom, and you will get offspring. And these offspring are extremely unlikely to already have hit the germline, or extremely unlikely to be already fully transgenic. Because that means that every cell of your blastocyst must have been transgenic, or one of those, well, for example, all cells of your liver, they all are made from a certain cell at a certain developmental stage. So if that one was transgenic, then all of your liver cells will be transgenic, and so forth. So until a certain stage of development, it's locked from where, I mean, ectoderm, endoderm, mesoderm, and so forth. You probably, or you should have heard that at some point, I guess. And what you need is the cells which you transfected need to be in the germline. Because then you breed those offspring among each other, and then you have a full generation, right? Or then afterwards, the whole mouse is transgenic. All right. I mean, that's not details. That's really basic principle. So next thing, spectral overlap. explain what it is and why this is a problem.
Spectral overlap is the phase where it is still absorbing the energy and at the same time emitting the fluorescence, and it is a problem because it can cause confusion.
Yeah, so first of all, what are we talking about? Is it cells, genes, organisms?
We're talking about...
You mean you're right? I just want to know what the material is that we're talking about.
It's a microscope thing. It's a fax.
It's a fluorophore.
Fluorophore, yeah.
So it's a fluorescent substance.
Yes.
It's very simple. If you use multiple fluorescences, or you detect at least multiple fluorescences, you can have the problem that you can, depending on your detectors, in one channel you can detect, or in two channels you can detect one fluorescence because it's a very broad band, but that's easy to compensate for. Or if you have multiple stainings, then one fluorescence can activate already the other one. So the emission of one fluorescence can be the activation, the excitation of the other one. So in this case, you need to plan the fluorophores and the detectors, because otherwise you will have signals. In both cases, you will have unspecific signals.
Can this happen with just a single fluorescence color?
With a single fluorescence, the latter you cannot have, because there's no second fluorescence that it can activate. So in a way, this is what we're doing when we do FRET. Then we deliberately want that.
Right.
Okay. Again, back to basic. Explain how a microscope works. So there's also this, all these...
Sorry about that.
Yes?
No, sorry. My Cortana activated spontaneously.
So all these questions are 3-point questions, because you can it's free for you how to address that, and it's not something where you write one word. Because I'm asking for principles, so you either have to draw something or explain, and that will take you longer than just making a cross at a multiple multiple choice question. So here in this case, explain how a microscope works. You can either write or tell, or you can draw something. So does anyone have something, what could be a potential answer, or?
I think here you can mention that there is an eyepiece lens, and I don't know if that would be enough to just to draw an eye, and then going down you can see the eyepiece lens and the objective lens and the source of light, and that how it manages to use the resolving quality of the objective to magnify.
Yes, exactly. I mean, we spoke about, for example, similarities between an EM and a light microscope, which exactly boils down to what you said. So you need some sort of, well, it doesn't have to be light, but you need some kind of radiation source. So if you want to explain a light microscope, then you should have a light source. If you want to draw a electron microscope, you should draw a radiation source.
Mm-hmm.
Yeah, so really, like, in the end, you want in your eye, which is a lens, you want to see what is in your sample. So in order to do that, you need light, which is coming from the light source or radiation, whatever, to hit your eye, and it needs to be in plane, so you need lenses. And with that, you also magnify and so forth. So that's a very basic principle. Yep. So key properties of a cloning and expression vector. What would you need? So first of all, what's the purpose of this vector?
To deliver the gene of interest into the sample?
Yes, but that could also be just maybe you mean it, but I'm not. It's not entirely clear. By deliver, that could refer to simply cloning, but there's another word.
You mean vehicle?
No, I mean expression.
Expression.
So cloning is just it's a vector that simply carries a gene, and you bring it into a cell. Expression means that the gene which you have in your plasmid is also then expressed. So then, what are features that you need on one of this vector? It should clone, so you have to carry a gene, and you should also express it in the end. What do you need to do that?
Mm-hmm.
I'm sorry, I didn't get that.
A promoter?
A promoter for?
Expression vector, I think.
Yeah, but to express what?
Well, in order to express the gene
yes.
that we've inserted into the vector.
This promoter needs to have a specific feature.
Origin of replication?
Yeah, that's for the plasmid, so that you can actually clone it, so that you can replicate it in bacteria. But my point is, if you want to express a gene, the promoter that you're using has to be adapted to actually work in that organism that you want to transfect. So if you use a bacterial promoter, it's never going to work in if you bring it in, for example, human cell culture. So your promoter needs to be working in the environment that you bring it. Or you use a promoter that works everywhere, like a viral promoter. What else do you need? An origin of replication. What else do you need?
Is it antibiotic resistant genes so that we can monitor the vectors which have been cloned or which have the gene?
Yeah, I mean, you need, so yes and no. Yes, you can use an antibiotic resistance, but you not necessarily, it must not necessarily be an antibiotic resistance. What you need is a marker or a selection marker.
Yes.
And then something like an antibiotic resistance is easiest. You could also go with a GFP, for example, or some other antigen that you can then detect. Or you could also overexpress, for example, toxins or antitoxins, or yeah. So you need a selection marker, and so we said we should.
Do we also need the cloning site?
Yes, yeah, that would have been my next point. So we said that the gene should be in there, so somehow you have to get it in. And in most cases, we're using restriction enzymes and ligases. So you need combinations that work. So you need a multiple cloning site. Yep, okay. So now we go to facts. This is a little bit more advanced, I would say. You have a mix of lymphocytes. You activate them, and then the B cells express CD28 and T cells express CD2. So you want to assess cell proliferation of B cells and of T cells. And I'm even nice and give you the molecule that you use to do that. So first task would be to describe the staining and FACS strategy that you can use. Do you all understand what you should do, or what's the scientific question, or what is the goal? What's the first thing you usually do in a fax?
Stain the cells, or mark them with antibodies.
Yeah, okay, yes. So let's assume that's already all been done. So now you're you're measured. So what would you look at the first? I rephrase. What do you not want to look at in your mixture?
You don't want to see the debris or the small stuff that you don't need. Like
exactly.
platelets.
And how do you exclude that?
I suppose the machine exclude that by setting up the parameters so that it ignores the small stuff.
Yeah, but how do you know what is small or large and so forth?
Oh, the side scatter.
Yes.
And the.
So what's the first thing? It's two things you look at: the side scatter and the
forward scatter.
And the forward scatter. So first thing is forward sideward. Unfortunately, I blacked out the questions now. Never mind. So we said you have a mixture of different lymphocytes, and you want to look at T cells and B cells. The antigens actually don't matter. So now you want to, so you pick here your, you exclude debris and you exclude aggregates. So you do forward scatter, sideward scatter. So then those cells, which you then transfer to another analysis, what do you do next?
You need to sort them out.
For what?
You need to sort them out for their sizes.
We've done that already.
Okay. For the characteristics.
Positive, negative.
Maybe for the B cells only and T cells only.
Yeah. So you want to know how do you define now what is the T cell and what is a B cell?
By attaching the primary antibodies with the different fluorophores, maybe B cells with the GFP or that's in the.
Yeah, so yes, correct. But that you've done that already. All of that is done. So the cells are already stained. So for example, you had a B cell specific stain, CD28, 28, for example. So what do you need to do now? You need to simply read your fluorescence.
You need to put it in, say, the institution and insert it in the analyzer.
I think it's probably just you're thinking way too complex. Your cells are already mixed with antibodies. Okay. One antibody against one of your antigens is coupled to FITs. Another antigen was coupled, or another antibody was coupled to another fluorophore, and another antibody was coupled to another fluorophore, and so forth. So you have lots of different colors. And previously we said CD4 plus, CD3 plus T cells. So there's two markers there and T cells. And even if you would not know that, CD8, CD3, and CD19 B cells, or CD28 once they are activated, and then CD2. So you want to know the T cells and the B cells. So you simply use, you stained already with the antigens or with the antibodies for those antigens. So now you read their fluorescence. So let's assume you had CD8, an anti-CD8 antibody with, for example, FITC, and a CD19 antibody with, for example, APC. So you simply read these two colors. Right, so you should get, your T cells should be very high in signal for FITC, and the B cells should be very high in signal for APC.
We could tag it with CD8 or also CD3, right? Because CD3 is in both of the T cells.
Yes.
Okay.
Yeah, so actually, that's a good point. You want to know all the T cells, so you should use CD3 instead. Because otherwise you will only have the cytotoxic T cells. Yeah? So you read CD3 and CD2. Here in this case, it's already CD29, it's already the activated B cells. You could have used also CD19, so there's always multiple options. So now you have cells which are in the upper left and in the upper right, so they are high in either of the two fluorescences, so these are your cell populations. But now we wanted to know whether they proliferate or not. And we wanted to use CFDA for that. Do you recall what CFDA does?
I think it binds to the element that's responsible for the proliferation, to the proteins that are part of the proliferation process.
Well, it's a substance that by itself is not fluorescent. Mm-hmm. Once it's cell permeable, so it can enter the cell. If it enters the cells, it will get modified by esterases so that it cannot leave the cells anymore, and it will also be fluorescent. So then you wash, and whatever is remaining in fluorescence is in your cell. So if cells divide, the level of fluorescence will shrink.
Yes.
So then you can read your cells that you selected before. You can read in the level of fluorescence in this. So maybe you go through the handout for these pieces. So there's other ways how you can monitor proliferation, but here since I said CFDA, this is how it would look like. Right. So also we talked about FRET.
One question.
Yes.
Can't we tag also the CD28 markers of the cells, like the markers that are expressed after the B cells and D cells are activated? I mean, and then look at the difference between.
The difference in what?
Oh, then that would be not proliferation. That would be activation, I guess. Sorry. I mean that the question said that there are some markers that are expressed when the B cells and T cells are activated, right? Like CD28.
Yes, that's.
Yes. So are we not using that information?
Oh, you did already.
But that was CD28.
Well, it's okay. It says 29. It should be 28.
Oh, okay. All right. Thanks.
All right. Blah 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 blah. Okay, here you want to use the FRET method to assess whether a enzyme, caspase 3, I guess you all have heard about it. It's an enzyme involved in apoptosis. Whether it's cleaving a certain protein. So you want to know actually you want to know whether a protein lysate that you have, whether it has caspase 3 activity. So you know that the caspase recognizes the sequence DEVD. So how do you design an experiment using FRET that you can detect this? So what's the principle of FRET?
I think the activation of one of the wavelength activates the wavelength of another fluorescence. It's kind of like
so you have two fluorophores, and the one activates the other. So exactly, you're using here the spectral overlap. And what is the key requirement in order from one fluorophore to activate the second one?
I think the wavelength has to be less than 10 nanometers.
So the distance.
The distance, yeah.
So they need to be really close. Mm-hmm. So how can you then, with FRET, with this principle, how can you check the activity of caspase?
If the chemical X interacts with caspase 3, like if they are close.
Oh, you can attach the parts of the proteins that once they dimerize. The idea is in my head. It's just like.
So you have two fluorophores, and they need to be close. How do you achieve that? So have you ever seen when, let's say, young kids at daycare, when they go out for a walk or something, and you don't want them to run away, what do?
They hold on to each other.
Yeah, so, or if you have a dog or whatever, what do you use? A leash. So what's your leash here? It's even written.
Probably the caspase itself. Or the peptide substrate.
The peptide sequence.
Okay.
So you link the two fluorophores with the peptide sequence. Because what happens if your tissue or whatever you use, you analyze, has caspase activity?
It's going to destroy.
It will cut in the peptide sequence, so the two fluorophores will lose their close proximity. So how would the FRET sensor or construct look like? I mean, you said it, just tell me, or draw one.
I'm so bad with drawing. I don't know.
Doesn't need to be artistic. It can be like lines and stuff. Just draw what you told me.
I'm sorry, I can't. It's still the ideas in my head. I think what I remember from the lecture is that, but I can't really recreate. I have to look into it. So, okay, so I wouldn't be able to draw it, sorry.
So, I mean, if you don't draw it, you can also describe it. But, I mean, this is, you don't need to be an artist to do that. I mean, I'm bad at drawing myself, but that I can do. Okay, so blah, blah, blah. So how will you perform the experiment?
Well, you need the radiation source to activate the very first CFP.
Yes.
Yes, and then I think you add the gas phase, or is gas phase already in there in solution?
So you have caspase 3, and you mix that with your sensor, which is a mixture of two proteins. CFP, YFP are proteins. You'll have them linked with the peptide substrate. If you do nothing, it will simply cut it. So now, if you add chemical X, it activates caspase 3. So caspase 3 was not active before. Now you add chemical X, so actually beforehand, the link was still intact. Now you add chemical X, so it will activate caspase 3, and then it's going to cut. So what's going to happen with the fluorescence, cis?
I think it has to show the peak on the emission 2 yellow fluorescence.
So what do you measure to actually calculate FRET? So what is FRET? How is it defined?
Ratio of fluorescence to signals.
Yeah, which ones?
Like exact which numbers?
No, but of DAPI, of FITSI, of rhodamine, of it's even written here.
Versus time. Okay.
So which one is the first fluorophore?
CFP.
CFP.
Yes. So what's the second fluorophore?
YFP.
So which one activates which one?
CFP activates YFP. If
How do you
there,
calculate Fred?
By calculating the amount of energy being used to expose
Not energy, you measure emissions. So you measure the intensity at certain wavelength.
Yeah.
So how do you calculate Fred?
By deducting it from the wavelength emitted by YFP.
It's a ratio, yeah. It's a ratio of emission 2 divided by emission 1.
Yeah.
So so YFP by CFP?
Yes.
Okay, so the higher emission by the lower.
Oh, yeah.
So first of all, what you do is you transfect cells with a plasmid that encodes your sensor. So you have it in the cell, so it's being made. Or if you've done that already, then you do it with proteins. But that includes a step that you actually have to purify this whole sensor, which is a lot of work. So you add chemical X, so you activate caspase 3. Of course, you should also have a control where you do not add it, your negative control. Then you monitor with the microscope. So that means you read the excitation of CFP, the emission of CFP, and emission of YFP, and that you either do at a time lapse or you do it at a certain time point. And then you calculate the Fred ratio, YFP, emission of YFP divided by emission of CFP. So how, now to number 3, how would the data look like over time? And here you would have to draw a graph. Actually, 3 graphs.
3?
1 for emission YFP, 1 for emission CFP, and 1 for the Fred ratio. Theoretically, you can also do it in 2, or theoretically in 1, but it's going to be, might be a bit distracting.
Oh dear.
Should CFP emission decrease over time?
CFP emission should
it decrease?
So if it decreases, then that means that the caspase activity would be more.
Okay. So it increases?
Less, sorry, it would be less. So then you would have an inhibitor of caspase, because if caspase is activated, it will cut, right? So you will see, did you say more or less?
I said less.
Right?
I said less.
So it will, because the caspase gets activated, so it will cut in between the 2 fluorophores. So there will not be a Fred anymore. Yeah. So you will see more of emission 1, meaning CFP.
Mm-hmm. Yes. And less of 2 emission. Okay.
And the Fred is simply emission of YFP divided by the emission of CFP. So in the beginning, it's going to be a very positive number, or a very larger than 1. They are all positive. But at some point, you will have a break even where it's going to be 1. And then with time, it's going to be less, because all the sensor has been cut. I think, yeah, that's the test stuff that I had. So as you see, I'm not interested in like that you simply put stuff in your head and don't understand what you do. But that means I don't want details and bull crap. So don't tell me the fluorescence maxima of CFP and YFP and stuff like that. But I want you to understand what Fred is. I want you to understand what an immune fluorescence is. I want you to understand what a microscope does.
Sorry, can you please explain, Professor, why I'm so confused right now? Like looking at the graph, I thought of something else. But why is the CFP is increasing in fluorescence?
So in the beginning, you have this sensor. So I don't know whether I can. Wait a sec. So in the beginning, you have this sensor, as it is here. So energy from CFP is transferred to YFP. So what you will read is mainly the emission of YFP. So in the beginning, before you've done anything, you will mainly read the yellow fluorescence. So now you give the substance X, and X is an activator of caspase 3. So it will cleave in here, meaning these two fluorescent proteins will leave their 10 nanometer proximity. So you will not see anymore the emission of 2. But of course, it will not cut right away all molecules. So it will take some time until the decrease is full. Yeah, so here half of the molecules have been cut, and here all of them have been cut. And vice versa, so here half have been cut, and here all have been cut. So the emission of YFP decreases, the emission of CFP increases, because now the emission of CFP is not anymore used up or absorbed by the YFP. Yeah, and since the FRET is simply a calculation of this value divided by this value, in the beginning you will have a large value because you will divide a greater value versus divided by a smaller value. So it will be a large one, and then you add your substance. It will take some time until it works, and then it will drop. So a reduction in the FRET value means that the proteins, there's no FRET anymore.
Mm-hmm. Thank you. Mm-hmm.
Okay, that's from my side. I actually also need to leave because there will be a lecture that I want to attend. Unless you have urgent questions, I would say good luck. Yeah.
Sorry, just one small thing. If to learn facts, not learn, just if you want to know more information about facts, would you recommend any books? Or the diagrams, like if we wanted to read more on them.
I would say any doesn't. I would say if you read a real dedicated facts book, it's way too much.
No,
I would suggest that you go and biochemistry books. They usually have method sections, or if you use something like Bruce Albert's molecular biology of the cell, stuff like that is enough. Or actually, my school biology book also had something on that, like already.
What is your school biology book?
I mean, as I said, I'm not going into details here. Yeah. So if you read Wikipedia, you will have the basic principle. I want you to understand what you actually do. Okay, I'm sorry. I unless you have something very burning, I would say we're done for today. And I wish you good luck with the exams. And I think, if I'm not mistaken, sometime in October, you should have the practical, if that works. But so maybe I see you guys then in the lab. So let's hope that that all of that works. We never know. So stay safe, healthy, and yeah.
Where is the lab going to be? Is it in the campus?
I think you're going to get all that information, but we are at the Botanical Garden.
Ah, in the UKSH.
No, in the near the biology tower. So it's am Botanischen Garten 11.
Ah, not the old botanical garden, but the new botanical garden.
Yes, it's at the Mensa 2. So you should have had a tour through the ZMB. That's where we are.
I see.
You didn't? Okay.
We still don't know where the university is. At least I don't know.
Yeah. Okay. You will get to know it in time.
It's here.
Right.
So I'm sorry I need to leave, but
Thank you very much.
Thank you very much.
Bye bye.
Have a nice
Thank
day.
you very
Bye.
much.
Thank you.
Bye.
Bye.